So as I mentioned today, we're going to talk about three topics: clusters, trajectories, and gene relationships. The first thing that we're going to go over is trying to talk about how different clustering algorithms work, to give you an understanding of sort of the biases that they might have. And as a sort of very first introduction to this topic, I want to point out that the goal of clustering is to partition the data space. So if we've got some set of features here, dimensions 1, dimensions 2, the goal of clustering is to create boundaries in that space. These are discrete, right? You're either in this region or in this region. This is called a Voronoi diagram, by the way. This says basically, like, if you had clustered this data again, if you saw a new point here, it would be in this cluster. We're seeing these partitions of the data space. And the goal is to understand how do you identify those partitions and what are some of the biases that you might find in different ways that you can create those partitions. So the first sort of question I think we want to think about is how exactly are we picking where to create boundaries between one cluster or another? There's a lot of different kinds of things that you might want. One of the things is you might want to have some objective criteria, like let's say you want all of the cells within a cluster to be close to each other. You have some similarity metric. You want to find the maximum split of the data such clusters are all similar to each other. You might want to, on the other hand, have separation. You might want to say, I want clusters to be far from each other. I want to maximize the distance between all clusters in the data set. You could think of a ratio between the two of them. We've talked a lot about graph-based methods over the past couple of weeks. One of the things you might want to think about is, are there sort of minimum sets of cuts that we could make across a graph, a nearest neighbor graph? We'll also, at the end of this section, talk a little bit about modularity, where you want to quantify on one of those graphs, are there some sets of edges that you want to see a high level of edges within a cluster and small edges between clusters. There's a lot of different ways you can think about doing this sort of clustering, and there's a lot of different objective criteria, and we're going to talk about a few of them today. So the first one, and the first algorithm we're going to talk about, is called KMeans. K because K is the number of clusters. And KMeans minimizes an objective criteria called mean squared error. You may be familiar with mean squared error. It's a general accuracy term used in machine learning models. It's also something that's used during fitting, say, logistic regression. You want to minimize the mean squared error between the observed values and the predicted values of your regressor. So the way that this works is you can imagine having some data set X, X1, X2, X3, Xn. Here are now observations of the data. We've used this notation a little bit to describe features. We're going to partition it into K different groups. And we want to identify these partitions such that within each cluster, there's minimum variance. And we can quantify this by saying, this notation here, some of you may not be familiar with. This big sigma means this is a sum. We're going to index from using this I variable from 0 to n. And we want to find the minimum for all means. Mu is usually used to denote means for each every cluster in the set of clusters. This notation means set cluster in the set of clusters. We want to minimize the difference between the points in that cluster and the center of the cluster. So the sum of all squared Euclidean distances between each point and that point's closest cluster mean. So that's our objective criteria. How do you actually go about creating partitions that minimize this, right? So I'm going to present to you now the algorithm. So by minimizing within such cluster sum of squared distances, we start with some unlabeled data set. And so that's shown here. We've got some set of X variables and Y variables. So this is maybe our two dimensions of the data. And just looking at the data, it looks like it exists in clusters, that they're sort of discrete partitions of the data. So the first thing I want to point out is that you can think of this data set and imagine creating 3 random centroids. These are the center points of the clusters. And so this is what we might call random initialization. You just pick 3 random points. The next thing that we do is we assign each point to the nearest centroid. So this is an assignment step. So there's initialization in step 1, and step 2 is assignment. So here what we're doing is for each point, we're saying which of these 3 centroids is closest, And then we're going to color that point based off of which centroid that was. So all of these points here are closest to the green centroid. All of the right-hand half of this cluster is closest to the red centroid. And then all of these points up here are closest to the blue centroid. And so what we can do now is repeat this positioning of assignments and then moving clusters to the average position in those clusters. So here we've got those assignments, and the third step, the next thing that we do is we actually take that green point and we move it to the center of all of the points that were assigned to that centroid. So here you can see that this red point has been moved towards the center of these red points. It's not actually the center of this cluster, it's the center of this half of the cluster. This blue point has been moved to the center of the blue points here. And green is actually a little bit farther to the right than the center of this cluster here, because there's some green points that we also want to minimize the distance to. So this is calculating the average, looking at the number of the size of the cluster. We want to minimize the average, or we want to find the average value of the X and the Y positions. We then repeat this until the algorithm converges or stops. And we talked a little bit about convergence when we were looking at magic on Wednesday of last week, right? This idea that at some point smoothing is going to see decreasing effects over time. And as you can imagine, this blue cluster here, this blue centroid is not going to move that much, repeating this algorithm, because all of the blue points are in this cluster. Once we've reassigned points to the nearest centroid and we move these points again, that blue cluster center is going to remain the same. The green one will move slightly to the left, the red one will move slightly to the left, but at some point they're going to stop. And so to show you what this actually looks like on some simulated data, I created a data set of 3 Gaussian distributions, and here we're looking at different iterations of K-means. So this is random initialization. So these centroids are randomly initialized. They're assigned to the closest centroid, and that's the color here. And you can see over time that this blue point is slowly starting to move towards the right. This orange point is slowly starting to move down, and that's just because randomly it turns out that the orange was a little bit closer to this blob here than this green one was. And over time, we start to see it around iteration number 7. There's maybe some differences in the cells here and here, but after iteration 7, the differences, how much the centroids are changing each iteration, drops off dramatically. And we can actually look at this. We can calculate something called inertia, which is the within cluster sum of squares objective criteria that we were talking about earlier. And so this is like this is the sum of squared Euclidean distances within each of the clusters. We want to minimize this. And what you can see is that right at about iteration number 7, the change in inertia at each increasing iteration drops down and eventually stabilizes. And so the idea is that at this point here, where there's now no more change in inertia over time, no more change of our objective criteria over time, we can say that the algorithm has converged, and you can then stop running the algorithm. And there's ways to look at basically the slope of this curve, right? We remember from calculus, you can calculate the derivative of this curve, see how much it's changing from one iteration to the next. You set a threshold. Once it has that slope is below the threshold, you stop the algorithm. So we talked a little bit before also about, are algorithms sensitive to their initialization criteria? And K-means is absolutely sensitive to the initialization criteria. One of the things that a lot of this sort of algorithm is known to do is converge to what we would call local minima, meaning that with this iterative algorithm, you'll come to convergence, but that doesn't necessarily mean you found the best possible stopping point for the algorithm or the best possible solution for the algorithm, but just the best possible algorithm given your starting position. So that imagine, there's another position where if you were to start from there, maybe your clustering results would be better. We would say that an algorithm that tends to find these sort of local minima is sensitive to initialization conditions. Now, I would suggest in some of the coding notebooks, we're actually going to look at clustering toy data. You'll be able to see on subsequent runs that sometimes the results of K-means is very different on different kinds of data sets, just setting different random seeds. And you can actually see and compare across algorithms which ones are more or less sensitive to that random initialization. There are a couple of solutions for K-means to figure out actually maybe a better way to create these cluster centroids rather than just picking like 3 random spots or K random spots in Euclidean space. One of the things, there's one heuristic, the 4G method, basically says, let's just take K random observations and make sure that those are our means, and that way at least you know that the initialized positions are really close to the data. Imagine here, there's nothing stopping if I just say randomly give me 3 points, all 3 points could be way off this XY axis. So what if we actually say, just give me 3 random points and those will be my centroids? That actually ends up giving you a little bit better results. There's also random partitioning methods where I actually just take random groups of cells. So say instead of just 1, say give me like 10 or 20, and then pick their average as a cluster centroid. So here's one question I have for you. I was telling you that the final solution I'm going to hide responses. Oh, sorry, turned off the screen here. Okay. Oh, shoot. Pull everywhere. Turned off. Give me 1 second. That's good. It means it's turning on. Okay, great. So 16. Okay, we've got 120 people on the call, so I'm going to wait again until we get about 40 people. Do you think that the algorithm will converge to the same or different positions? Interesting. We've got about 50-50 split. This algorithm will always arrive at the same solution, or no, it will produce different solutions given the same initialization. And so I think that the algorithm that I presented earlier, this idea of taking the centroids, assigning them. So if you're given with a set of initial states, and then you assign points to those centroids, I hope what you'll see is that that step is deterministic. There is always, if you're given a centroid, the set of points that are closest to that centroid are always the same, right? Because Euclidean distance, when you're calculating the distance between one point and another point, that doesn't change based off of some stochastic principle. It's always the same. The next step in the algorithm, in this iterative algorithm, is moving the centroid to the center of all of the points that that cluster centroid was assigned to. And I hope that you'll see that this step too is deterministic. If I give you a set of 100 points in Euclidean space and you want to calculate the average position of those points, that is a deterministic step, right? So you're always going to see the same mean. If you just say, take the average, the numerical arithmetic average, that will always be the same. So that step is deterministic. And then the next step, right, we're recursing now, or not recursing, we're iterating. We're then reassigning points to their closest mean. That step as well is deterministic. And so because this is an iterative algorithm composed exclusively of deterministic steps, calculating means, okay, one person did make a good point in the chat, which we did not talk about, but is true. In the case that you have 2 points, let's say you have a point, you want to assign it to a cluster, and it is exactly equal between those 2 clusters, you have what is called a tie, and that tie is solved randomly. I would just like to point out that that is an implementation detail that is not inherent to the algorithm, how you decide to do that. It would be equally likely that you could say, what if I don't want to solve the ties randomly? What if I just always want to say, I'm going to assign that point to the cluster with the smallest value, the smallest average or something. You can imagine a deterministic way to break ties as well. But yes, it is true that if you decide to break ties randomly, that this would happen. But at least the steps of the algorithm that I presented earlier are all deterministic, meaning that this is a deterministic algorithm, meaning that you'll always find the same solution given the same starting condition. So the fact you get different solutions with different starts doesn't necessarily mean that the algorithm will give you different ends given the same start.
I just wanted to just relate again. So we have talked about another greedy other greedy algorithm. So TSNE and UMAP, which we talked about in the visualization part, are greedy, so they're heavily dependent on their initializations. And in fact, I told you that a lot of the performance improvement in UMAPs comes from the fact that they initialize with a Laplacian eigenmap. So just something to remember, that if somebody's running gradient descent, that's usually also a greedy algorithm. And we'll talk more about gradient descent tomorrow when we talk about neural networks.
Okay, great. So I want to point out, so this is one algorithm. This is probably the simplest and most common algorithm for doing clustering in arbitrary data. Like in a machine learning perspective, if you said, give me clusters, I think the first thing a lot of people would try is K-means. But K-means has a number of assumptions to the algorithm that I think are very important to understand and understand the point that this is not necessarily going to apply to every data set. If these assumptions of the algorithm are not met, that the output is not going to be reliable. So this is some points taken from, there's a really good blog post called K-means is not a free lunch, lists a number of assumptions. If you're interested, I would recommend reading this. But there's basically, there's a few ideas. So one is that K is chosen correctly. So here we have what I would call the incorrect number of blobs case, where you have 3 blobs. And someone asked before, what do we mean by a blob or Euclidean blob? What I mean is it's something that looks basically like this, like something that's random, normal, or uniform in a circle, but something that's like basically round and equidistant from a center, or some variation of that. It doesn't necessarily have to be exactly even in all dimensions or directions, but that you have some sort of even distribution. So one is that you have the right number of blobs. If you say, give me 2 clusters and the data actually has 3 different populations of points, these clusters are not going to be correct, right? That should make sense. The second one is that you assume that the data is distributed normally about the mean. So not just distributed normally, but also normally and with even variance. And so there's 2 points here. So one is that these might be normal in each direction, but that the variance of each of the dimensions is different. So we call that anisotropic because they don't have the same variance. The variables don't have the same covariance is basically what this means. And so here there is a high covariance, or they're not independent. There is a covariance between variables. And so here, these sort of round or long blobs do not get clustered properly because they're not round enough. There's also an assumption that you have equal variance, so that basically the average distance from the center of the cluster to all of the points is the same across all of the data sets. And so here, if we have 3 different clusters, this one is the largest and has actually a really high variance. What you can see is that when we're clustering, actually some of the points that should be from that cluster are actually getting called as part of this blue cluster because it assumes that they're all going to have equal variance. And that there's also the same number of points in each of them. What if we have a data set where there's imagine these are cell types. There's 2 common cell types and 1 rare cell type. K-means is not going to do a good job of picking out that rare one. And so what you can see is that, in this, we're talking a little bit more like what happens when those those assumptions are violated. We could have a data set, what I was showing before, that you look at the data, you could see that there's probably 3 clusters, but then you cluster and you get the wrong ones. And so the question now is, what do you do? And this is going to get us now into the beginning of our second clustering algorithm that we're going to talk about today. So the next one I'm going to talk about is called spectral clustering. Spectral clustering was introduced actually is a NeurIPS paper in 2002 by Andrew Ng, who you might know as being responsible. I think, did he help found Coursera? I know he has one of the most popular machine learning courses on Coursera. And
Yeah,
he's
I think he was one of the founding members along with maybe Dr.
Daphne Cole.
Fuller. Yeah.
Yeah, and he also, I mean, a really big name in the field, went on to lead Baidu, which is one of the big search engines in Google, and actually introduced a way to do this kind of clustering based off of a graph. And so one of the messages, we really like to hammer home in this workshop is that graph representations are really useful for data. And a lot of the methods that you're learning about rely on this idea that a graph is a useful representation. How do we understand that? Here's an example. Here's a data set. And the fundamental idea for building a graph is you take this nonlinear function kind of kernel matrix that takes distances and makes them affinities, and then you can build these connections between points. So here you're actually looking at basically a graph on that data. This should look kind of similar to that manifold learning introduction notebook from last week. So I'm going to talk to you now a little bit about some of the matrix representations of that graph. Some of these we've talked about before, but I really want to make sure that we're reusing a lot of language and you can sort of see connections across different algorithms. So one of the fundamental matrix representations of the graph is called the adjacency matrix. And the adjacency matrix basically just says it's a square matrix, so it has as many rows as it has columns, and the entries in that matrix are the edge weight between nodes I and j. So let's say that we're looking at this point and this point. There's a very faint line here. There's a very weak affinity between these 2 points. And so we'd say that maybe the adjacent, the affinity there, the edge weight is like 0.1, for example. But then between this point and this point, the edge weight may be 0.2. And so as you look along that affinity matrix, you can see those edge weights. We looked at some representations of this last week. So that's one thing that we can look at. The second one is the degree matrix. And some people have asked a little bit about what do we mean by degree. And degree says if I look at a graph and I have all of the edges going out from that point. So there's 4 edges going out from this node in the graph here. What's the sum of their affinities? So you could imagine if you were doing a k-nearest neighbors graph, all points might have exactly the same degree. Each point is connected to its 5 nearest neighbors, and so they all have degree 5. Excuse me. But in a weighted graph, like what we get with the kernel matrix or kernel function I showed you earlier, you might get different values. Some points are more connected than other points are. And this gives you some information about that connectedness. How central is it? High degree might mean it's really central. So a point in the center here is going to have a lot of connectedness. And then there's now finally a really useful matrix called the Laplacian, which is the degree minus the affinity matrix, plus some normalization. Sometimes you normalize this in a certain way, so that either maybe the rows are, it's perfectly symmetric, or it's always symmetric, but the rows sum to 1, or there's a few different kinds of things you can do here. So there's this Laplacian matrix. And what I want to convince you, we talked a little bit about eigenvectors last week. What I want to convince you is that eigenvectors of that graph Laplacian are really useful for clustering data. And we talked about this a little bit in the idea of Laplacian eigenmaps, right? We're looking at the eigen decomposition of a Laplacian matrix. You can plot those, and then you can actually visualize the data. But one of the things that was recognized in this first spectral clustering paper is that actually this provides a basis for clustering. So what do we mean by that, by a basis for clustering? So typically, when I was showing you K-means earlier, the basis, the input to the data, input to the algorithm was a matrix of n rows by n genes. And then you would just cluster based off of that. But there are other ways that we could do this, right? You could imagine taking the first 100 principal components and then clustering on those. So in this case, the 100 principal components is the basis that we're using for the algorithm. But principal components are linear. We showed earlier that Laplacian eigenvectors are nonlinear. And what happens if you have data that actually has some cluster structure to it and you build a graph, here we're showing the data and the color represents the loading of the Laplacian eigenvector onto that point. So the 0th or the first eigenvector is not meaningful, but the first through k+1 are. And what I hope you can see here is that eigenvector 1 really well separates this small cluster from the rest of them. Eigenvector 2 has really high values in this cluster. and low values in this one, and sort of middle ones here and here. And the third one really just isolates this point. It's actually kind of far away, so it's maybe not as useful, but what you can see is we can then make a new representation of the data where this psi1, psi2, and psi3 represent the same eigenvectors we're coloring here, right? These, now we're just saying that's going to be the x-axis, the y-axis, the z-axis. And what I hope you can see is that these separate the clusters quite well, and I'll show you that a little bit in a little bit later. I hope that it's clear that as you go up above the number of clusters that you have in the data, that the eigenvectors become less meaningful and more localized. So here, actually, even potentially just 2 eigenvectors would be really good for partitioning the data. And typically, you might want to ask, how do we pick the number of eigenvectors? The answer is you usually pick the number of eigenvectors as you have clusters, as you want clusters, because this is used as an input to K-means. And so we see here now, you cluster on this space, we actually get really great segregation. Splitting this eigenvector 2 in half does a good job of splitting green and blue clusters, and then splitting eigenvector 1 in half really splits the orange from the rest. And so this gives us now clustering that better fits our data. And we can look at that shown on the original data axis to see now that spectral clustering, I'm just saying spectral K-means, but this is usually called spectral clustering, does a better job of handling one of those cases we were talking about earlier, where you have an uneven number of points per cluster. And so actually, we tend to use spectral clustering first when we're looking at a new data set and we want to identify new cluster components. We have shown a little bit earlier, we were talking about these ideas of Laplacian eigenvectors, and I want to show you that these are meaningful across different kinds of data sets. This is just now saying, what if we have 4 clusters and we want to get 4 different clusters out? We can see here that maybe the first 4 that we're looking at are going to be useful, and above that they start to get more localized. Here even maybe again we only need 3 because this graph is very regular.
I'm going to connect back to a prior lecture again just to jog people's memories.
Please.
So remember that when we talked about Laplacian eigenmaps, we did talk about the fact that these eigenvectors are the same as the eigenvectors you get in diffusion maps. Except for in diffusion maps, you weight the eigenvector by the eigenvalue. So we had also previously shown that these eigenvectors light up separate components of the graph, and therefore that method of dimensionality reduction is better for something like clustering than for visualization, because each eigenvector is sort of lighting up one component. That's the same thing that's being shown here, that these eigenvectors disentangle the components in the data space, and therefore it's an easy representation to cluster on.
Yeah, I agree. I think that's a good way to explain it. I also want to show that you can look at these eigenvectors regardless of whether or not the data actually exists as clusters. This is a pretty famous example for graphs. This is Minnesota, and this is a graph of all of the roads. You can't see them here because there's a ton of them, but between every adjacent intersection in this data set, there's an edge. And we can look at eigenvectors of this to identify different kinds of partitions of the data. And so you can imagine maybe if you split on the first eigenvector, you get the top half from the bottom half. The second one's going to identify the Minneapolis metropolitan area. The third one sort of gets this bottom part and the top part, separates out each of them, right? So you're going to get clusters here. And you can sort of see that the more clusters that you want, the more resolution is given using higher resolution or more eigenvectors, the more splits you're going to get because they get higher frequency. This is actually from that initial publication of spectral clustering. These are the data sets they showed you could isolate using this technique. You can look at letters, weird different shapes. There's a lot of things that spectral clustering actually does really good at resolving. These are just toy examples. I also want to touch base really quickly. I'm realizing I'm actually going to pick up the pace a little bit because I want to make sure we have some time for the other 2 topics today. But some people might be interested in understanding how do we pick the correct number of clusters. And I'm not going to actually go through this section right now, but I want to just get across the idea that there's a metric called the silhouette score, which looks at basically the difference between intra-cluster distances. So this is, remember that inertia we were talking about earlier. This is sort of what K-means is looking at. We want to minimize intra-cluster distances, and maximize inter-cluster distances, right? And so you can go back through the slides later and see exactly how this is calculated. But what I want to show you is that if we look at a function of this ratio between inter and intra-cluster distances, we want to maximize it, right? Because we want the highest, we want the highest difference. We want, or it's a minimum actually, but I'll say that we want to minimize the intra-cluster distances and maximize the between cluster distances. And you can actually plot this using many different numbers of clusters and say, well, when do I get the highest ratio? And that might give you a suggestion of how you pick K. This is not the only way that you can pick the number of clusters. There's a number of different techniques, but they all work in essentially this way. You formulate something, desirable quality. I want to minimize this and maximize that, and just take sort of the average difference between the two. And then you can actually evaluate the algorithm using different sets of parameters and say, for what set of parameters, K being the parameter here, do I maximize that objective criteria? And this gives you a way to pick K. I talked a little bit before about spectral clustering being a graph-based clustering method, but spectral clustering doesn't necessarily take into account explicitly let's see, it does take into account the graph-based nature. I don't know the best ways to say this. It doesn't take into account some graph qualities other than the representation of the graph. But having a graph, there are different kinds of attributes of that graph that we can calculate and then run cluster.
You're going towards community detection here?
Exactly.
The only difference is that community detection doesn't use the K-means optimization criteria.
Yeah, I'm realizing actually
optimization criteria.
Yeah, I maybe just shouldn't have included this slide. Another graph-based clustering criteria is clustering by modularity or community detection. And if you've heard of Louvain or Leiden clustering, this is probably one of the most popular algorithms. It's implemented in 10x Cell Ranger. It's implemented in ScanPy and Seurat. The idea is to cluster based off of modularity. And the idea of modularity is explicitly trying to quantify the number of edges within a cluster, or they call it a module, compared to the number of connections between them. So here, this blue, there's a lot of edges between them, but there's only one connection to the teal cluster and one connection to the green cluster. And we see in a random graph where there's no modular structure, there's just a bunch of connections within and between. And so what we want to do is actually create partitions that maximize this number of internal to external. I will explain, I'm actually a little bit hesitant to spend a ton of time explaining the notation here because I think it's more important that we get to pseudotime. But the basic idea, I hope you'll go through these slides and try to understand. One of the goals I have is, this is, I think, one of those intimidating sort of formulations of an objective that if you don't know what all of the terms mean, makes it a little bit difficult to approach. But I've annotated these slides with basically a guide and then also sort of a simple interpretation of each of the terms that I hope will help you understand a little bit of what we're actually looking for. And so the basic idea is we want to look at the connectivity between nodes within the cluster, adjusting by the average number of edges per node, that degree we were talking about earlier. Because the idea is that if you have a high degree, you're going to have more edges within the cluster. And so this allows us to calculate a statistic called modularity that we can then iteratively assign points and partition the graph such that, or assign points to clusters, and as a result partition the graph such that we can optimize this Q measure here. And I hope, looking similar as those graphs earlier, that this graph on the left here, where nodes 1, 2, and 3 are assigned together, 4, 5, and 6 are assigned together, that this has higher modularity than this graph does. I think actually, if you want to understand how that algorithm works, like in the same way we were talking about that assignment and then moving the means for K-means, there's an improvement to Louvain clustering called Leiden clustering. And actually, the paper that describes Leiden clustering does a really good job of explaining how Louvain works and then why some of those decisions of the implementation don't actually function as well as they could, and then describes improvements to that algorithm. I think this is a really good introduction to that topic. And it's also written, I think, for not necessarily, it's more approachable than some other algorithm papers. One of the notebooks we're going to look at later today is this toy data set. I was talking a little bit about this. We're going to look at a bunch of different kinds of data and then a number of different kinds of algorithms. And we're going to see basically, so each of these, each row is data set, each column is an algorithm, and you can see, so this is all implemented as part of scikit-learn, how they perform on different data sets. So, fundamental ideas for clustering here is that we're partitioning data to identify groups of similar observations. K-means is an iterative algorithm minimizing within cluster differences. We can also use a change of basis, looking at the eigenvectors of the graph Laplacian to perform a similar task. Or we can start to look at measures of community and modularity to perform tasks like Louvain and Leiden clustering.
I just wanted to recap that. Can you just go back to the previous slide, Dan?
Sure, yeah.
One thing that I would say is that from my experience, clustering is a lot more subjective than I think most biologists attribute to clustering. Most biologists seem to have an idea that there are these cell types out there and we simply have to discover them using the right algorithm. That's not true at all. Cells don't maybe naturally exist in our types. It's a construct we're putting onto the cells. And so it's an extremely subjective method. The optimization criteria you use, whether you should cluster or not, how many partitions you pick, what are you going to do with them? Those are all extremely subjective things, and it's a choice you make. So sometimes when somebody tells me, oh, there's K clusters in my data, that has much less meaning than a lot of people think it has. It depends on how they partitioned it, why they did it, what criteria they were using, all of this stuff. So hopefully you can take away from this that there's many heuristics for this, but no sort of fundamental ground truth.
Awesome. Thank you, Smita. I guess, I'm sorry, this slide got a little bit chopped off. So the question here at the top is, how do we identify differentially expressed genes between clusters? This is a question that has come up a bit in some of the Slack and office hours talk, and I'm going to just acknowledge this very briefly. Let's say we have a data set that's partitioned into clusters. We can start looking at histograms of the variables, right? So this is x1, this is the x dimension here, and this is x2, right? So we're looking at the y-axis. So this histogram shows that. What I hope you'll see here is that x1 has much higher values in that cluster versus the other two. And here, this cluster 1 has much higher values for dimension 2 than the other ones. And what I hope that these remind you of is from statistics, the idea of comparing two sets of distributions. So here we're looking at two normal random variables, and we want to look at the overlap between them. We want to look at the distance, the difference between the means, and the variance between them, or the standard error of the data sets. And based off of how close or far away these are, we can calculate a statistic called the t-statistic. This is an example of the t-test, right? The null hypothesis is that the means are none. The alternative is that there's some delta between the means. And then we can use sort of the variance to tell you, based off of, or the estimated variance of the random variable, using the data, sort of how confident we are that that distance, that difference is statistically meaningful, right? So you could, in this data set here, this data is all normally distributed, very valid to do a t-test to calculate those kinds of differences. And actually, it's not that uncommon for people to do this kind of thing, to actually look at a t-test for single cell data. However, I want to point out, let's see, I'm actually just going to skip through this, that there are other solutions for when the data is not normally distributed, right? So we know that single cell RNA sequencing data is not normally distributed because there's this huge dropout problem. We actually have counts, right? We're counting things and we know we're not counting all of them. So it's more of like, imagine you're doing a survey of a population and you know you're only getting 10% of the population. So there's a lot of zeros we're going to see if you didn't ask some people some questions, for example. But there are other I just want to mention that there are other kinds of tests for this case as well, right? So one of them is the Mann-Whitney, or sometimes called the Wilcoxon or the rank sum test, where instead of just looking at the difference between the means and the variance. We actually look at the ranks of the values for each of the genes in rank of each of the cells across the two samples for each of the genes, which gives you some non-normal solutions. You can then look up the statistic. So, there's normal tests, there's non-normal tests, but the thing I actually really want to hammer home is that these tests also have their own set of assumptions. And one of the ones that is common to both of them is that all of the observations are independent from each other. So this is the first one in both. Actually, this is just, I must have deleted something. This should be highlighting the first assumption here as well. So they both need to be independent. But one of the problems is that because we have clustered the data, the two clusters are not independent, because you cannot be both in one cluster and another cluster. And the clusters are almost always defined based off of one of the variables that you're going to use for differential expression testing. So imagine that your data was, so this is 500 samples of a normal distribution, right? Imagine that this is the gene. And then we cluster the data. So you get one cluster here and one cluster here. So this is from a cell systems paper from 2 years ago, but we cluster them, and you always cluster based off of the values. So by definition, because you're partitioning the data into high and low, just like we were doing here, right? We're partitioning the data into high and low in order to get these two clusters. We've now enforced a model where the data is no longer independent. And because of that, the p-values tend to be artificially inflated. So this is just a histogram of statistics for the t-test, but there's basically any different test you do, the p-values are inflated. And so this paper actually introduces another test that estimates basically how inflated it would be based off of a bunch of random partitions of the data. I have not really seen this being used that much. People don't really do it. I think the reason is that even that test that they're doing has a bunch of different kinds of assumptions. And at the end of the day, what I think a lot of people have failed to think about is that they're very obsessed with this idea of p-values. Is it significantly different or not? But that I just want to make sure that people understand that sometimes what's more important than knowing is it statistically significant is just how different is it. So we call this a measure of effect size versus a measure of significance. And so even if the p-values are meaningless, it doesn't mean that your estimate of the difference is not. And that might help you just ranking genes that you want to look at based off of full change, or the rank sum or the t statistic, even if you don't necessarily want to trust the p-values. So with that, I'm going to stop talking about clustering. I really would encourage anyone who has questions to stay after in the main session after the lecture. But I'm going to dive right into pseudotime and trajectory inference. So we talked a little bit before about what happens if you have data sets that you think are somewhat discrete. Maybe you have T cells and B cells and NK cells and eosinophils, and you want to isolate those different populations. Clustering is a really great strategy to do that. And one of the things people talk about, like, how do you know the right number of clusters? Oftentimes you can just look at a data set, you look at some markers of genes that you know are going to be in there. You have peripheral blood cells, you know what they're going to be, and then you can very easily segregate them. But in some data sets, you have continuous changes, and actually clustering is not the right paradigm. You want to understand, how do cells continuously go from one state to another? So you can imagine a really simple developmental system where you have one axis here, where we're looking sort of, genes that are changing as you go from state 1 in purple to state 2 in this tan color. And dimension 2 is basically just noise. And you might want to know, like, what are the genes that are expressed in each population? What genes change the most as you go from early to late? Like, which genes come on early in that transition? What genes come on late in that transition? Clustering is not really going to help here, because clustering doesn't actually give you an order. What I hope that you understand is that if you wanted to answer these questions, the first thing that you need is an inherent ordering of this developmental process. So you want to order cells from early to late. So you can imagine doing an experiment where you have some cells in growth factor and you measure them at different points in time. Or sometimes you actually don't even have to do that. If this system is naturally distributed across all of these states, you could do this all with just one sample if you knew the beginning and end point. And you get some set of days that you're measuring. The goal is actually to identify this underlying progression. You'd like to reorder them such that cells from state 1 are the earliest, and cells from state 2 are the latest. I'm going to talk a little bit in this section about how we actually identify that ordering, but the key phrase is that pseudotime produces an ordering of cells. So pseudotime, also sometimes called trajectory inference, because you can imagine trajectory sort of like a path from early to late. There are actually over 70 methods that are now compared in this Dyneverse data set. This is one of the most heavily, I don't want to say maybe investigated, or just there's more solutions to this problem than I think for many other problems. Like there's, I think, more trajectory inference methods than there are clustering algorithms, especially in common use. And it's really important to understand that they each have their own set of biases. We're going to look at one of the first methods, diffusion pseudotime, and then actually one of the top ranked methods, which is slingshot. And so diffusion pseudotime here is one of these bifurcation methods, actually doesn't perform that poorly, and then slingshot, which actually performs quite well. And I want to explain to you how they work, and so you can understand a little bit about what they're doing. So imagine we have a system like I was showing before. This is actually simulated single cell data using splatter. You can imagine, identifying data from 3 different time points. There's a ground truth axis of differentiation. I know this ground truth because this is simulated data, and I said, give me data that follows a trajectory, and then it generates this and gives you this label. And then you can also assign it to different days. And I'm going to bring back an idea that we talked about way earlier, this idea of graph distances. And I hope you'll understand, we sort of introduce some of these fundamental concepts and explain to you how they can be used in many, many, many different kinds of algorithms. So before we were talking about graph walks to approximate manifold distances. If you have 3 points, 0, 1, and 2, and you want to know which ones are the closest, you really should walk along this graph rather than just taking distances in the ambient space, right? Because you might make jumps across this where there doesn't really exist. So we have some way of basically taking walks along the graph that gives us some measure of distances. And what I want to point out here is that actually just that simple idea, saying what is the graph walk distance from, say, let's say we just knew, because this is a stem cell system maybe, and you know what stem cell markers are, that this is the earliest point, right? Maybe you looked at a fate graph of the data, or you looked at PCA even, and you notice this is the earliest time point. We're going to say this is the root. You can just look at the graph walk distance through the data set from this point to all other points and get out a pretty good estimate of that ground truth differentiation trajectory, right? So this is just an ordering. It says, how far are you along the graph from that first point? So this is essentially the same idea as diffusion pseudotime, but instead of just looking at specifically graph walks, we look at diffusion distances, which simulate many, many, many walks, and say, how long do you, if you were to take 10 steps, how likely were you to be at one point or another? So you just ask, how do you determine the earliest point? I will first of all say, great question for the Slack. But I'll address now again, imagine that we assume that you know something about the biological system you're studying. People usually don't just pick up a single cell data set off the street. Usually you are studying a system that you sort of understand. And so it might be stem cells, for example. And so maybe that there's a stem cell marker like OX4, and you see expression of that is really early here. And so you select it. We're actually going to give you a task where you can do this in one of the notebooks today. You can actually get some experience picking that earliest point. It's literally, this is exactly that question. It's the next slide. I forgot that. So you might see, for example, that there's some differentiation marker that you know of. Maybe you know marker for a terminal cell point, and you see that it's lowest at this point. And so that's your starting point. So you have to use some expert knowledge to guide the algorithm. The reason for that is there's no way to know just looking at this data set, is this the start, or is this the start, or is this the start? You need to constrain the algorithm. Otherwise, there's no way to solve this problem. So diffusion pseudotime I talked about simulates random graph walks to calculate those distances from a start cell to other cells. So it doesn't just look at the actually diffusion distance from the start or the graph walks. What it actually does is it looks at the difference. It calculates each point based off of its diffusion distance to all other points in the data set. So this mx comma dot is saying, I want to know the, imagine the Markov distance, the random walk distance, the diffusion distance from one point to all other points. And I want to calculate the difference, the diffusion pseudotime distance between 2 points is the difference between these random walk vectors. And so if that makes sense, if you look here, this is our graph walk distance. What I hope you'll see is that so this is the walk distance from every cell from this cell. What I hope is somewhat clear is that let's say that red cell was this point, the color, and then you colored by distance to that red point, the graph wouldn't change very much, right? The colors would be very, very similar. The graph distances are going to be almost the same. And so those diffusion pseudotime differences are going to be almost zero. And then the rest of this is actually not that important for you to understand. What this term says is that they perform this analysis over essentially every possible set of random walk lengths. So right, we're talking about the probability that after 10 steps you'd be at this point, but what about the probability at 11 steps and then 12 steps or 100 steps? So actually this creates a series, and the bottom part of this provides what's called a closed form solution, or a numerical, I guess not necessarily numerical, but or a computational solution that allows you to explicitly calculate the product of this series. And you may be somewhat familiar with infinite series, and they have limits, and you can calculate their limits in some cases. If you know of like Fourier or Taylor series, they're guaranteed to converge to a certain value. You can calculate what that value is. I don't think I really learned about how to do this. The last time I talked about this was in like senior year of high school. So it's not necessarily as a biologist, applying these methods, you don't necessarily need to understand exactly what the math is, but that idea of a limit is important. There's one other point I just want to make super quickly here, which is that this framework also provides a way to detect branches. And I'm not going to spend a ton of time talking about it, but what I want you to just understand is that there is a special relationship. This might be one endpoint in the data set, and this is another, and these purple cells are the beginning. Paths from X to Y and Y to X, and then from Z to X has an inverse relationship that as you're going on this branch from X to Y, the diffusion distances are getting smaller, but actually the distances to Z are getting larger. And this might be a little bit confusing right now. This is actually also covered in one of the coding notebooks for diffusion pseudotime. But what you can actually look at, there's this idea of correlation, anti-correlation at bifurcations that provides a way to from a data perspective, identify where the branches are. And if you're really interested in that, again, please, if you're interested in understanding how you can detect branches using diffusion and pseudotime, come to the main office hours and I can talk about it a little bit more. But the basic idea, what I want to drive home here is that having these orders, this ordering of cells, provides a way to answer some of those questions that we had before. So let's say we just had the 3 days. This is, again, simulated data, so I know the ground truth. I have a set of genes. They're just randomly numbered here because it's simulated data, and they don't assign fancy gene symbols. You can just say gene 1, gene 2. Here we're looking at 4 genes, and we're looking at their average expression at day 1, day 2, day 3, or and what you can see is that there's not a ton of differences for some of them, right? Like here, it's like high and then kind of doesn't really change between 2 and 3. This is always increasing. This is always decreasing. This is always increasing. Well, what's interesting is once you actually get this diffusion pseudotime value, we can look at the graph walk, this ordering of cells, and actually some interesting things happen. Like I bet that you wouldn't have guessed that this really went down and dropped down in between days 2 and 3. But pseudotime suggests that a little bit over halfway through this trajectory, this gene goes down and then goes up. Or that this gene starts high, goes low, and then goes up in between these 2 samples. Just looking at the days themselves doesn't give you a way to do this. And what's really cool is we can actually know, so this data set has ground truth. We know how these genes are supposed to change throughout this process. And so we can confirm that the ordering that we're getting and the gene trends we're identifying along with time are valid. Right, so we can simulate data and use that simulation to just say that this kind of pattern could be picked up in this context. Sets diffusion pseudotime, right? So that is a context where we're talking about graph walks and diffusion distances to calculate an ordering of cells from beginning to end. So I said this is one of the first methods to be described to do pseudotime inference. It's not the first. Monocle is one of the first ones. Actually, Cole Trapnell's lab coined the term pseudotime, but diffusion pseudotime sort of fits, I think, a little bit better within the framework. What we're teaching is important to understand. So what about the methods that score the best? How do modern methods that really sort of perform very well? Okay, so first of all, I said modern, right? But I want to point out that we're going to talk next about an algorithm that uses a tool called principal curves that was actually generated in 1989, this first idea of principal curves. And this, I think, is a really useful plot to understand at a high level of what principal curves are doing. So at first, I want to just mention we have two concepts, linear regression and PCA. So linear regression, we were talking about before, the goal of linear regression, let's say you have some data set here, this is your x-axis, this is your y-axis. What you want to do is identify the minimum error, minimize the residuals along the y-axis between the line that you're identifying and the prediction of that value, right? So linear regression says if I have a value of 0.5 here, the expected y value is here, but actually maybe the value I'm getting is here. This is pretty close, that's fine. Here, if it's at 0.1, I would expect the value is going to be here, but actually the observed data is very high. So this is a high residual, high error. We want to minimize those. This is actually along one of the axes because you have to specify what a predictor is. But I want to contrast this to PCA. PCA could be, we talked about PCA as an eigendecomposition of a covariance matrix, but you can actually reformulate this in a different way as minimizing the orthogonal distances between all of the points and a line. What is the line that has the minimum difference between all of the points and that line? So you can imagine actually a whole new algorithm that is just defined as doing this. It doesn't actually look at eigenvectors of a covariance matrix, which is how PCA was defined, but it gives you very similar results. I will point out, actually, this is a very similar thing to the difference between UMAP and t-SNE. There's a very fundamental relationship between the two, but a reformulation of how you're approaching the problem might give you a new algorithm with a new implementation, where that implementation is actually is much better than the original in some ways, faster or gives you better things, but fundamentally they're very similar. And actually, I want to point out that this way of talking about orthogonal distances is a similar way to talk about PCA. So regression along the y-axis, PCA orthogonal to the line. Orthogonal just meaning there's right axes. So there's also nonlinear regression. And one of the ways that nonlinear regression works is saying, what if I just want to locally fit lines? So I'm just going to say, let's take the first 10 data points, give me a line that minimizes the error along the y-axis between that line, and then I take another section and I create another line, and then I locally create these paths or these components of a curve that minimize axis and the error in the y-axis. And this is the lowest algorithm, if anyone's familiar with this. This is a similar way where you're just looking at specific parts of the data set that gives you now sort of a non-linear path through the data. Principal curves do a very similar thing using this orthogonal definition. So here we're saying, let's identify a curve through the data that minimizes the orthogonal distance between each of the points and that curve. So it's similar sort of into PCA, that's where the principal part of the name comes from, but it's a curve because it's non-linear. You want to identify a non-linear path. So you imagine this is similar to saying, what is the curve that explains the maximum variance in my data set? Noting, however, that there's no orthogonal curve to any given non-linear curve, right? So you can do an orthogonal to a line, but not orthogonal to a curve. So this is just, you can get multiple curves through the data. So Slingshot was developed in 2018, and the way that this works is first you run this algorithm on a dimensionality reduced embedding of the data, say 2 or 3 principal components. I know that that's low, but that's what they do. You can imagine 5 or 10. In the notebooks, you can play around with this. You could also use Fate as a basis for Slingshot, which I think we also are going to do in one of the notebooks. The second step is to cluster the data. So actually, we're taking a continuous thing, and now we're clustering it using K-means or spectral clustering. and then creating what's called a minimum spanning tree between the data sets. This is actually, minimum spanning trees are very similar to the first pseudotime algorithm that was developed. But here, basically, you would imagine is these are the centers of each of the clusters, and you want to identify connections between them that has the shortest amount of total edges in that tree. Note, tree and graph. All trees are graphs, but not all graphs are trees. And so this is a tree representation of those clusters. And the goal is to fit principal curves to that tree. So there's one curve for each branch on this tree, because you actually identify branches here as well. And then we project data onto those curves, right? These are those orthogonal residuals. And actually then this ends up being the point. So this point actually might get a little, although it's further to the right in the feature space, might actually get pushed a little bit to the left because it's orthogonal than this point. But this actually works really well. And you can see these steps shown on a biological data set where we have data that gets clustered. You create the minimum spanning tree in the data domain, and then you fit curves to the data and project. The projection onto the line gives you that pseudotime. We're going to do Slingshot and Diffusion pseudotime in the coding notebooks. I think this is the last thing I'm going to talk about in this section of the lecture today. Is another way to start to think about transitional dynamics. So those are pseudotime methods. What I hope is a little bit clear here is that this is just using the geometry of the data or the shape of the data in either the ambient or dimensionality reduced space to identify paths through the data. And then you can look at these paths to identify how things are changing along them. One observation that was made a couple of years ago in Peter Chekhenko's lab at Harvard was that in a lot of different kinds of single cell sequencing methods, even ones that were supposedly biased towards looking at the 3' end near the poly A tail of the gene. So for those of you who aren't familiar, this is the 5' end of the gene. These are exons and introns. I'm saying it's mostly for the TAs who maybe come from more of a math background. And this is called the 3' end. Most of the single cell technologies are geared towards getting this 3' end of the gene. And there's usually a poly A tail here that they pull on a set of recognized nucleotides. But so typically the gene has already been spliced when you get an RNA and an mRNA to be polyadenylated. And so this middle section here, these are called introns, should be excised. But one of the observations that they made was that actually a significant portion of reads contained splices, contained the introns. They were unspliced. And one of the things that we know is that the way that RNA is made within a cell is that you have DNA that gets transcribed into pre-mRNA or unspliced mRNA that contains these gray bits. These are those introns. These are these middle things that are supposed to get removed before you have spliced mRNA. And it is spliced mRNA that is used to create proteins in the cell. And then that spliced mRNA is degraded at some rate. So here, the Greek letters denote rates, right? So there's a rate of transcription, and rate has to have a denominator of time. But you can imagine, let's just say we're looking at rates over a 1 second interval. So the rate might be 1 transcript per second. That produces some quantity of unspliced mRNA in the cell that then gets spliced at some rate. And in this paper, they assume that there's a constant splicing rate, which then leads to a concentration of spliced mRNA, which then is degraded at some rate to get sort of destroyed and recycled within the cell. So these rates are really what we would like to estimate. What if instead of just looking at the data geometry, we could actually see what genes are getting turned on or have active transcription, and what genes are getting turned off, which ones have active degradation. And so the idea is we can't actually measure those rates because you have a static snapshot. But because we actually get some of these unspliced and spliced RNA estimates in each cell, we can actually use those quantities that we measure. These are quantities and not rates, and describe a differential equation that expresses the amount of the unspliced and spliced RNA as a ratio of the degradation rate, which gives us some measure of a ratio, which gives us some measure of how actively transcribed or degraded the gene is. And so the idea here is that if you have more, so if you, let's say you knew the degradation rate, which which they estimate across a whole data set, you want to know, is the unspliced greater than the amount of spliced times some degradation rate? If there's more unspliced than spliced, the idea is that you have active transcription because this step is happening faster than this step. If you have equal spliced to unspliced, then there's nothing happening. You're at steady state. The cell is just, you imagine like actin, for example, probably is equally turned on and turned off, or transcribed and then degraded. But then there's also a repression regime where the rate of transcription is less than the rate of degradation. So this is a gene being turned off. And so you can actually look at the ratio of unspliced to spliced RNA to find out where you are in this space. And there's a number of really cool experiments that they did. One of the first ones was bulk RNA in mouse liver. Actually, I think that they didn't do this. I think that they analyzed the publicly available data. But here, this represents each of a bulk time course. And you can see every 3 hours they collected measurements and look at this ratio for different genes of the spliced to unspliced ratio, and can actually recapitulate circadian rhythms in the liver, which are known to, which have been previously observed. And if you actually look at PCA representation of each of the samples, this cycle is observed within the data sets just based off, and the arrows here now give you an idea of what is the future state going to be. Because let's say that this gene is turning on, you could estimate at some time point later, say an hour later, that that gene is going to have a higher concentration in the cell. So it actually gives you a vector. We're not just measuring a snapshot, we're seeing a snapshot and a prediction of a future state, a direction of transcription or a direction of degradation. And one of the powers of this is now we can actually describe data geometry just based off of the mRNA content of each cell, looking at a projection of the data here. So this is from the RNA velocity paper. We can look at a projection of the data. We can look at the diffusion endpoints. This is using a thing like diffusion pseudotime. They know that these are the induced pluripotent stem cells. And so this is the, actually, I'm going to take that back. I'm not entirely sure I'm right on that expansion of this abbreviation, but they know that this region is sort of the beginning time point. They see the ends are sort of at the tips of this embedding, and that we can then look at the arrows that say, for each cell, where is its future state going to be, and that they point towards the end of the data set. So not only does diffusion pseudotime suggest that these are the ends, but also the rates of transcription and degradation give you this idea. And we can see this actually what's really cool is we can maybe imagine having 2 points in this data set. These are 2 different cells. They're very close together transcriptionally. But if you look at their transition probability based off of RNA velocity to other cells in the data set, this cell looks like it's actually going to go towards this upper branch of the data set. And this cell, which on a global transcriptional level is very similar, is actually going to go down a different path. And so the idea is that this is going to be an entirely divergent fate that you would not have predicted just based off of the mRNA expression of the cell. But by looking at the ratio of spliced to unspliced RNA, you can identify this. With that, I'm going to end the pseudotime inference section of the lecture today. The key takeaways from here are that pseudotime and trajectory inference aim to identify an ordering of cells that matches some developmental or intrinsic progression. I want to point out that this could not necessarily be a developmental progression. It could be that you have disease progression. It could be that you actually have a drug titration experiment and you want to know what happens as I increase the dosage. It's not just day 1, day 2, day 3. It could be dose 1, dose 2, dose 3, and identify a progression that way. You don't need multiple time points for pseudotime because oftentimes this relies on you saying where the beginning and end are of a data set. 2 of the methods we talked about today use either graph walks or axes through the data to identify those orderings, and you can find comprehensive comparisons on Dyneverse. I know this is a ton of algorithms that we've just introduced. This is like, we've now gone through 6 different algorithms in just over 60 minutes. And so I would really encourage anyone who has questions to be active on the Slack or come and chat with us in the office hours. Happy to talk a lot more about this or other trajectory inference algorithms. And with that, I'm going to hand things off to Smitha, who's going to handle the last component of the lecture today on learning gene-gene relationships. I don't know, Sveta, do you want to request control of the screen here?
Yeah, so I did put the Slack on the Slack a reference to the comparison paper that Dan had referred to. There was a question there about how somebody would go about deciding which method to use. I think the general answer is kind of understanding your system and how that method works and what it might be sensitive to. So, for instance, we did talk about how diffusion distances are quite robust to noise. Some other ways of constructing trees or paths through the data may be less robust to noise, but they might be more sensitive in some cases. So these are the kinds of trade-offs. So now I'll be talking, just a quick note again. So Alex will be talking about optimal transport-based methods that talk about how to do something like pseudotime, which is find a progression when you have gaps in your data. Because the pseudotime methods are really best for one, they're basically marking an axis through a single manifold. So if you had wide gaps in your data, you'd want to do something like called optimal transport, where you're actually transporting data points. And that has been a trend in single cell analysis. There was the Waddington OT cell paper by the Aviv Regev lab, and our lab had a paper called Trajectory Net and ICML, and Alex will be mainly going over those tomorrow at 3. So I'll be talking about sort of the final topic of this lecture. So the theme of this lecture is sort of extracting characteristics about your data, after you've pre-processed it, batch normalized it, and visualized it. A pretty common kind of post-processing technique that people in biology want to do is clustering, or sort of cell type delineation. More recently, there's been a trend towards understanding that data can have continuum structures, and so you might want a trajectory or ordering. There is another major type of quantification or information you can extract from single cell data that's sometimes overlooked, and that's the idea that you can learn about relationships or trends either between genes or along axes of progression, for example, along pseudotime axes. And often the single cell data itself and the techniques I'm going to talk about are a starting point, and you can use other methods to kind of prune the networks that you can use, for example, with motif analysis in single cell ataxy. So just the idea here is that you have these single cell measurements where you have simultaneous measurements of different genes. So because we're measuring something like 20,000 different genes, especially after you denoise your data, for example, with MAGIC, you'll see how 2 genes vary together because you have simultaneous measurements in many single cells. And this is something that you actually don't have at all in bulk data. And so the idea here is that if you're looking to see what is the relationship between gene X and gene Y, each single cell is a point of information about this relationship. So if you've measured tons and tons of cells and you start to see this kind of relationship emerge, which means that in the cells where gene Y is highly expressed, gene X is lowly expressed and vice versa, then you can infer that there might be an inverse relationship between these. And one of the ways that sort of MAGIC is useful is before the noise, you might, by random sampling, not have measured the same gene in 2 different cells, same set of genes in 2 different cells. But when you recover and denoise the data, it might become apparent to you. And often these relationships are not like the inverse line that I just showed you. They can often be nonlinear or have a variety of shapes. Now, people have often told me that all the meaningful relationships in biology are linear. And you'll hear people in various fields saying that. To me, it seems very unlikely. The reason is because biological entities have heavy regulation. For example, you can only produce a certain amount of a gene after which it can be capped, which leads to nonlinearities. There's often a lot of feedback as well. So when we're really looking at single cell data, we often do tend to see nonlinear relationships like this one. And so I'll be talking about how to detect these kinds of nonlinear relationships. Here's a poll EV question. How do you know that the second pair that I showed you in the previous slide might have a relationship, but not the first? Is this poll actually working, Dan?
Yeah, it looks okay. Yeah.
Mm-hmm. I have trouble going back and forth on these slides when you share.
Yeah, it's not the. Part of it I think might be the Wi-Fi connection to my laptop is not great. I don't have an Ethernet cable for it, but
Yeah. Um, Okay, so a lot of these answers are good. The idea is if you can predict what y is from looking at x, you have an idea that there might be some relationship between them, whether it's interaction or co-regulation by the same transcription factor. A lot of you are saying correlation. Correlation is biased towards picking one kind of relationship. Um, and graphing is correct, but I'm talking about when you look at a graph, how can you tell if there's a relationship or not? Usually, when you have a relationship, there'll be some sort of trend. It doesn't have to be linear or nonlinear, where there's a definite change in the y when x changes. Whereas in the first relationship I showed you before the denoising, it's hard to detect that relationship because it looks like there's no relationship because the way that y is changing is kind of random when you look at how x is changing. So the intuition behind mathematically detecting relationships from single cell data or hypothesizing relationships, whether they're direct or indirect in single cell data, is looking to see how what's on your y-axis changes when your x-axis changes. So if you want the change to have a certain form, for example, if the change has to be proportional, then you're looking specifically for a linear relationship. If the change is in a specific direction, so if it's always a positive change, then you're looking, for example, for positive correlation. But often these plots are all from real biomedical data when two genes can clearly have a relationship. Y is changing when x is changing, but it neither has to be proportional to the change in x, nor does it have to consistently have the same kind of direction. If you use correlation as a measure, correlation is very related to covariance, which we were talking about. It's actually a sort of normalized covariance between two variables based on the standard deviation of each of those. And you see that correlation is very good at picking up these kinds of relationships that are basically linear. If you have a noisy linear relationship, the correlation starts to go down. If it's negative linear, it's fine. Whereas if you have a relationship that's wildly nonlinear, you might miss it. And if you use correlation as a way of extracting these potentials for relationships, you might miss it. So one of the techniques that we go over here is mutual information. So mutual information is actually a powerful way of detecting potential relationships between genes that comes from information theory. It's basically asking do two variables have information about each other? I know the word information is used a lot colloquially, but there is actually a very specific mathematical definition of information. Information is actually defined as the amount of uncertainty that's reduced when you know a particular variable. So if x has information on another variable y, that means that knowing x reduces the uncertainty of y. And the question is, how do you measure this uncertainty? One way of measuring uncertainty that we've talked about before is variance. But variance has some specific problems. So in information theory, the amount of uncertainty is measured as something called entropy. Entropy is actually measurable as a quantity called bits. So for example, you might think of a fair coin as having 1 bit of information. So this is the same bit that you talk about when you're looking at your computer and things you represent on the computer. The idea is if you want to record the result of a fair coin flip, then you need 1 bit to record because you can encode heads as 0 and tails as 1. So that only needs 1 bit. But if you're recording the result of 2 coin tosses, you can no longer do it in 1 bit. You need 2 bits now to do it. So the amount of information is measured basically as the number of bits you need to more or less write down the result. And it's measured given by this formula. If it's the probability of each of the outcomes times the log of the probability of that outcome. So if you have a fair coin, the amount of entropy in the fair coin is half times log of half, so negative half times log of half, and then another one. And if you add these up, you get 1, and that's how you measure how much entropy you have. So information is basically going to be asking, how do we reduce the amount of entropy? Is knowing information about the one variable reducing the entropy in the other variable? And so here's an example of a relationship that actually has high mutual information. The idea is that you can quantify the uncertainty in y. That's basically the spread of y. So y itself, think about just projecting all of this data out to this axis here, has quite a bit of entropy. But if you know what the value of x, let's say you can narrow down the value of x to this slice, then the amount of entropy in y is accordingly lowered much more. And this is actually what mutual information is measuring. Whenever you know the value of x, the range of y is actually very small. So knowing x reduces the information in y. And this that idea that you have a different amount of entropy when you know another variable is called conditional entropy. So it's the entropy of y when you condition on another variable x. So the conditional entropy of y given x is the average of the amount of entropy you have in each of these bins. So on average, you're going to reduce the amount of entropy in y by quite a lot by knowing x. So just a couple of things about conditional entropy is that the conditional entropy is necessarily less than the overall entropy because you can't increase entropy by conditioning because all you're doing is slicing it. At the most, you won't do anything. So mutual information is just the difference in these two quantities. It's the difference between the overall entropy of y minus the conditional entropy, which is the average entropy in each of these little bins here. So this is actually estimated just like this using these kinds of bins. So if you have continuous data, you would bin it into a certain number of bins. You compute the entropy of y by binning the y, and then you look at the entropy of x. So just to give you a little bit more intuition, you can see that certain kinds of trends would have low mutual information. This has low mutual information because, to begin with, the y doesn't have much entropy. It's always taking on the same value. But knowing x isn't really lowering that. It's just the range of y sort of staying the same. Similarly here, this is kind of a noisy trend. It has some amount of entropy. But if you sliced it with respect to x, it's not directly decreasing it. Whereas these kinds of relationships, even if whether they're monotonic or non-monotonic, do have high mutual information because the overall range is high, but the range in any one x bin is low. So this is why mutual information is able to detect a wide variety of relationships, unlike correlation, because it's just talking about is the uncertainty reducing. To compute mutual information in actual data, what you would do is you would take the overall range of y amongst all your cells or data sets, and then you can divide it evenly into a certain number of bins. You can decide the amount of bins, and then you would do the same with the x, and now you have these little squares that are xy bins, and you can recompute the entropy in each of these columns. So the bins of y, just binning y, which are these horizontal bins, help you compute the entropy of y. And then the entropy of x given y is an average of the entropy in each of these columns, given these y bins. And then you compute, and that gives you mutual information. Mutual information is always a positive quantity, so you can rank how much mutual information you have amongst things. People in computational biology have used it for network learning, particularly the papers from Andrei Califano's lab. They've also used another quantity called this is called the data processing inequality. So usually they're also trying to see if there's direct or indirect relationships. And to some extent, you can also do that with mutual information. If there's a direct relationship, the idea is that X has less information about Z than Y does, because Y is an intermediary. But conditioning on a third variable can actually increase or decrease information about a variable because of this reason. One of the things that we noticed when we were first trying to apply mutual information to this is that imbalanced data is a problem for mutual information. Usually in gene-gene relationships, you have most of your cells or genes concentrated in some location. So most of them are in this point of the relationship. So you're overemphasizing this part of the relationship as opposed to the whole relationship to see if there's actually a trend between these two, regardless of the cell they're in. And this would mean that you're really looking at like a small portion of the entire curve. So instead of looking at this whole relationship, you're looking at just that, which might not tell you much about the mechanisms as well. So in order to address that, back in my postdoc, I actually developed this measure called DREAMY, which is actually a density resampled or reweighted in the way that it's implemented estimate of mutual information. The idea behind this is that cells in lowly populated parts of relationships are useful for unveiling relationship structure. And the way DREAMY does it is it actually takes this joint probability and computes what's called a conditional probability estimate. So here you have data. You can compute density of the data. You can compute density of the data by dividing the data up into bins and seeing the probability that it falls into one of these bins. And then the joint probability is just the probability of cells falling into any of these bins. This is called the joint probability. Usually when people compute mutual information, they go off this joint probability. So these are the bins that I talked to you about. You've got the probability in the bins and you apply the formula. But this kind of thing, makes you really ignore these sparse area. Instead, what is done in DREAMY is these values are all column normalized. So you get this conditional probability. It's saying if the x is here, the y lies over here. And now you're starting to get an idea of the full relationship. Now, if you take this set of bins and these values and compute mutual information off of it, you're going to be scoring the whole relationship rather than just the dense area. And that's the idea of DREAMY. Now, Dreamy has been used in order to try to learn relationships. For example, transcription factor target interactions. In the MAGIC paper, we use the epithelial to mesenchymal transition. MAGIC definitely helps the Dreamy scores. So you have things like this where you're not detecting any relationship, and after MAGIC, you start to see what the relationships look like. And if you do too much of it, it might devolve the relationship, but the right amount of MAGIC tends to help it. So usually what we do is we have data after MAGIC, and then this can be fed to Dreamy. We have a package called KMeans Dreamy that does it. Another thing that you can do is you can try to figure out if relationships are dynamically changing over pseudotime. So you have these cells are ordered by pseudotime using potentially one of the pseudotime methods that Dan talked about. And then you can compute mutual information with respect to pseudotime. So here also you see that there's a lot of nonlinearity. A transcription factor like snail in this epithelial to mesenchymal transition process, it's going up somewhere. So let's look at snail. It goes up and then comes down again somewhere. Whereas this one is up first and then it's going down. So you see these different kinds of relationships, and we're able to reason about when in the process these genes are involved using this as well, this form of mutual information with a pseudotime variable rather than two variables by themselves. And just to show you an example, when you do Dreamy, you can get low scores on things that don't seem to change alongside each other, and high scores on things that have very distinct thin relationships. So that's something you want to watch out for. If the relationship is thick, there's a lot of uncertainty there. It's probably intermediate regulators. If it's very thin, there's probably a strong relationship. So the conclusion here is that mutual information can be used to identify nonlinear relationships between variables, which seem highly abundant in single-cell RNA sequencing data. And the density normalization in Dreamy allows us to quantify mutual information even in this presence of highly imbalanced data, which we have using these technologies. You can use it as a way of identifying drivers of differentiation or gene-gene relationships, but this is also an association statistic, so you need corroborating evidence to actually infer causality. So things that can help you are things that give more evidence. So if you see gene-gene relationships using Dreamy, then you can look at other things like chromatin accessibility or actual knockout experiments, and this will bolster your confidence in these methods. But it is a good start given that you have 20,000 relationships or 20,000 squared types of relationships in your data. It's a good start to start with this kind of a network. Any questions?
Great.
I'm going to quit sharing.
I mean, yeah, so that's the end of the lecture for today. And so thank you, Samantha, for giving that part of the discussion. I want to, this concludes, I think, marks a switch in sort of the topic matter for the workshop. A lot of the first 4 days really focus on graph-based manifold learning methods. The last 2 days of the workshop talk about neural network-based methods. One of the reasons that we think it's really important to talk about neural networks is because there are a lot of new technologies and techniques, algorithms that are using neural networks, and especially for large scalable analysis. There's a lot of like fundamental ideas here that we think are really important to understand. And I would really suggest, if you've never heard really about neural networks, you have some fundamental ideas about what they are, I would highly suggest that people look through the first couple of chapters of these two books about neural networks. The first one, Neural Networks and Deep Learning, is I think much more of like a beginner level for people in the beginner track who maybe have never heard of this before. Someone just asked, we're going to cover graph neural networks, and I don't think that we are. I would have to double check this.
I could add a couple of slides on that the last day.
Yeah, so maybe that's a good idea. If there's other, I mean, if anyone has suggestions, stuff like that, me or Smitha, we can, touch on some of those topics. Tomorrow it's gonna be much more of a basic introduction, and the second day is gonna talk a little more about some specific algorithms and very sort of more detailed material. So the second one I would recommend people look at is this deep learning book. There's also on the workshop materials website a bunch of learning resources, including videos on neural networks. I did not understand neural networks like probably the first 3 times they were explained to me. And so I think if people here wanted to spend at least just like a little bit of time watching a couple of videos, trying to understand how they work, hopefully the lectures will be a little bit more approachable. And so I guess actually this, we don't need to talk about this. That's leftover from last year. So with that, we're going to end the lecture component of the workshop for today.